Hello everyone, my name is Deane and welcome to my presentation. Today I'll be giving an abridged version of my thesis defense and talking about my research from the past few years. The title of my presentation and my thesis work is Metabolic and Physiologic Strain Differences in Response to Constant Light in Male Mice. Circadian rhythms are the biological clock that are responsible for maintaining many of our body's daily functions. The light cycles we're exposed to every day are the regulatory driving force to this clock. Daily entrainment is important for good health because it's a regulator of many of our hormone cycles. However, disruptions in this cycle have been known to lead to or exacerbate metabolic and behavioral problems. Both human and rodent studies have shown that jet lag and night work can lead to the development of diabetes and cognitive defects. One form of circadian disruption that is becoming increasingly common is constant light exposure. This can include night shift work and the use of TVs and smartphones at night or throughout the night. This prolonged exposure to light at night alters your circadian rhythm. Previous studies have shown that constant light can lead to altered metabolism, including diabetic symptoms and disruptions to hormones in animal models. Several studies have also shown that night workers, not genetically predisposed to having diabetes, end up developing diabetes because of this exposure to light at night. In addition to metabolic issues, constant light can also produce increased anxiety, stress, and depression-like behaviors in both human and animal studies. The basis of my research is to understand how different strains respond to constant light exposure. The C57 BL6 mouse is a very commonly used inbred strain of mouse in research. However, two distinct colonies in this strain have been created, one at Charles River Labs in Massachusetts and another at the Jackson Labs in Maine. These colonies have been separately bred for so long that it has resulted in the formation of two distinct C57 BL6 substrains. The B6N mice reside at Charles River and the B6J mice at Jackson Labs. For many years, these strains have been considered interchangeable until recently there has been evidence to suggest that there exists several strain differences in the way they respond to circadian disruptions. This creates variability in many study results. My study was done to define differences between these two genetically similar strains of mice. For this study, I took half of the mice from each strain and placed them into constant light, while the other half remained in normal lighting conditions. From there, each group was also divided in half again and placed into cages with access to a running wheel or a cage that monitored activity via an IR beam. These cages are also two of the most commonly used, so it is important to define any differences in the home cage types. Both cage types allow for the tracking of circadian rhythms. To outline strain differences in response to constant light, I performed the open field test, which measures exploratory behavior in a novel environment, which gives the mouse an opportunity to explore in an open cage, the light dark box, which is a similar setup with half of the cage covered and measures anxiety, and finally a novel object test, which measures recognition memory by performing repeated exposure to either a familiar or a new object. I also performed a glucose tolerance test to analyze glucose metabolism, and lastly, I performed a liver triglycerides molecular assay to establish the fat metabolism of these animals. I found that in a novel environment, B6J mice increased distance traveled compared to B6N, but running wheel access reduced distance for the B6J mice only, and this is regardless of the lighting condition that they were in. So this happened both in light dark here and in constant light here. The B6Ns in LL exhibited the greatest center zone time amongst all of the groups, so that's these two light gray bars here. In a normal lighting condition, B6J mice exhibited increased light zone time compared to B6Ns overall, but decreased in constant light only for the B6Js. B6J mice and constant light both had influence on increasing the number of transitions between the light and the dark zone. So you'll notice that these variables altered this transition, but independently of each other. Lastly, in the novel object test, we see differences in the interactions with the first object between the B6Ns and the B6Js the first time that they're experiencing the object, but we see a dramatic decrease in B6N mice, but only in IR cages regardless of the lighting conditions, and this is with the right object, so it's with the new object. Home cage locomotor activity was higher in the B6Js, in the light-dark animals, and in the running wheel animals, but they were all influencing running wheel activity independent of each other. All mice were able to entrain to the light-dark cycle, although we see period lengthening as expected in constant light. In LL, B6Js had the longest period lengthening, and this was regardless of cage type, and running wheel axis exhibited shorter periods than the IR cages in constant light here. B6Ns in constant light gained the most amount of weight compared to the Js in constant light, as well as the Ns in a regular lighting condition, but this was only the case for the IR cages. And lastly, we see an increase in liver triglycerides in both strains, but only in the IR cages, and this is again regardless of lighting condition. After seeing the differences in the other two strains of mice, we wanted to see if these differences were caused by the disruption in their circadian cycle or if it was through the light input. So the second part of my work looks at the CBAJ and the CBA-CAJ strains of mice. These mice are genetically similar to the previous part, with one main exception: the Js are blind and experience retinal degeneration. Again, half of each strain was placed in a normal lighting condition of 12 hours of light and 12 hours of dark, while the other half was under constant light conditions. Mice, primates, and humans have a harder time synchronizing if they are blind, but they are able to synchronize to their circadian cycle if the retina is intact. There is evidence to suggest and support that there are photic and non-photic receptors that helped us with entrainment. Circadian light receptors and visual light receptors are both located in the retina, but go to different parts of the brain, and this gives us some overlap of information, but is not complete overlap because they are two distinct pathways. The cages generate a graph that looks like this. On the 12th day, the animals going into constant light were placed into that cycle. This is where we see the line start to slope. And this is the free running period of the animals. So we can see that it is longer in the mice that can see. Their slope is a little bit shallower than that of the blind mice. So we see a stronger circadian dysynchrony in the mice that can see. I found few interactions with the CBA strains, and the differences were always seen in the mice that could see in constant light, because the blind mice responded the same whether they were in a normal light cycle or if they were in constant light. This pattern is seen in active time, so we see the largest difference here in the mice that can see in constant light. We also see this difference in glucose metabolism. This is the calculated area under the curve for the glucose tolerance test. And again, we can see this big difference in the mice that can see. And the last, our big difference, is our average weight gain, in that the biggest difference is caused in the mice that can see in constant light. The first part of this work revealed several baseline strain differences. So under normal lighting conditions, under a standard light-dark cycle, B6J mice exhibited increased locomotor activity and reduced exploratory behavior compared to the B6N mice. In constant light, however, B6J mice have greater period lengthening and increased anxiety, while B6N mice exhibit increased weight gain and experience no change in exploratory behavior. The B6J mice also decreased exploration with running wheel access, while the B6N mice did not experience this difference. The results from this first part reveal and demonstrate that the B6 substrains exhibit different behavioral and physiological responses to circadian disruption as well as to running wheel access. The second half of my study revealed that constant light does not affect the blind mice as strongly as the mice that can see. The blind mice do not experience the same period lengthening as the not blind mice, which is a clear photoperiod and retinal function difference. The amount of light intensity to the brain increases the photoperiod because even with the slight overlap of photic and non-photic cues to the circadian pathways to the brain, it does create some differences because of the incomplete overlap. Because the mice that can see were more susceptible to the circadian disalignment of constant light, they were the strain that gained the weight because of the lighting cycle. In combination, I was able to replicate past constant light results such that constant light was found to alter novelty-induced activity, and we showed changes in metabolism and thyroid function, and this was independent of the strain. I would like to thank the Adrian Tisley Program, the Office of Undergraduate Research, and the Biology Department for helping fund my projects, as well as a huge thank you to my mentor, Dr. Joseph Seggio, and to the BSU Seggio Lab for all your help with these two projects. Thank you all for listening to my presentation. Feel free to comment any questions you may have. Thank you.